Hello, welcome back. We're now going to discuss some background to the field of proteomics, which is something that a lot of people haven't really learned much about, because I think that it will help you to understand how we can measure the final product of gene expression. So let's start with the definition of the field. Proteomics it encompasses a very large number of disciplines, and so I'm going to just use the Human Proteomics Organization's HUPO's definition for what proteomics is. It focuses on the identification, localization, and functional analysis of the protein makeup of the cell. The proteins present in a cell, together with their function, subcellular location, and perhaps even structure, change dramatically with the organism, and the conditions faced by their host cells, including age, checkpoint in the cell cycle, and external or internal signaling events. So clearly, it's not just transcripts that are moving up and down in expression. Proteins are too. Where proteins are located within the cell, how they're structured, both in terms of which modifications they're carrying and how they're folded, impacts their function. The most dominant pipeline that we see used in proteomics right now is sometimes called shotgun proteomics or discovery proteomics. So I'm going to try to walk through this in a bit of a hurry, and then we'll come back to talk about mass spectrometry, and more specifically tandem mass spectrometry, in just a minute. So we start with mixtures of proteins. These may just be a total cell lysate, for example, thousands of proteins in a mix. We then will digest those proteins to work with peptides, and the reason is principally that peptides are easier to work with analytically, and they're easier to identify from the data we can collect from them. We separate those peptides for very complex mixtures. Maybe you have 100,000 different peptides that might be measured in a cell. We separate those to fractions, and then each fraction gets subjected to a technique we call LC-MS-MS, and that's what the rest of the diagram here is. So we start with liquid chromatography, which separates peptides based on how hydrophobic they are, more or less how greasy the peptides are. We electrospray them, and we'll talk about that in a following slide, to create ions from these, because if it's not an ion, we can't measure it in mass spectrometry. Then we have two different kinds of measurement that take place within a mass spectrometer. We have a high-resolution mass measurement that is just trying to ask, what are the masses of ions, the mass-to-charge values, that's what we actually measure in a mass spec. What are the masses of the peptide ions that are currently available from the ions that are flowing out of the liquid chromatography? Then we can isolate a bunch of ions of a particular mass-to-charge value of a particular peptide ion, bounce it off of a bunch of gas molecules, which causes it to fragment, and then measure the set of fragments that were seen for a particular peptide. And that's called a tandem mass spectrum. Now we're going to revisit a lot of these steps as we move, so I hope that this helps you to understand each step in detail. First off, one of the key things that I mentioned before was that we must have an ion if we're going to measure something. There was a Nobel Prize given out that encompassed both the process of electrospray ionization, which is coupled to liquid chromatography, the method we use for separating peptide mixtures. And there was also, and the same Nobel Prize was awarded for MALDI, matrix-assisted laser desorption ionization. Essentially, you have a gold plate and a little spot of proteins embedded in matrix. You hit it with a laser, and up pops a plume of ions. So both of these cases are useful for biological mass spectrometry. The first when you use liquid chromatography, and the second when you don't. Okay, now a mass analyzer is one of the essential components of an instrument. So we need to have ionization to produce ions. We need to have a mass analyzer to selectively stabilize ions. And we need a detector in order to evaluate the amount of a particular ion that we have. So the time-of-flight mass analyzer is probably the easiest type of mass spec, and also a great many others: Fourier transform instruments, ion traps, quadrupoles. It's a huge display of different kinds of instruments. But the time-of-flight mass analyzer, I think, will help you to understand how ions can be separated with respect to their mass-to-charge ratios. So let us imagine that we've used the MALDI technique to produce a plume of ions. This plume of ions now need to be separated in order of their mass-to-charge values. So we accelerate them all to the same kinetic energy. And if you remember your physics, you remember that the mass is part of this kinetic energy. So if you have accelerated a disparate collection of ions to the same kinetic energy, the small ones are zipping by, and the large ones are moving more slowly, because that's how kinetic energy works. So we can time how long it takes for the ions to move from point A to point B. And if we know the distance from A to B very, very accurately, we can infer what their velocities are. That velocity, in turn, gives us the mass value, the mass to charge value that those ions have. As they hit the finish line, so to speak, they're hitting a detector. And this is sort of like an electron multiplier. The arrival of an ion kicks loose an electron. That electron kicks loose more electrons, and those electrons kick out more electrons. So, in effect, the arrival of an ion is going to produce an electrical signal that increases if there are more copies of that ion or decreases if there are fewer. Okay, so let's move ahead. Tandem mass spectrometry is not just asking what ions are flowing out of the liquid chromatography or from the spot of the multiplate at this moment in time. Instead, tandem mass spectrometry represents a structural interrogation of those peptides. So by colliding the peptide ions, now we have a whole bunch of copies of one peptide, ideally, in the instrument this time. The mass analyzer causes the many copies of that peptide ion to collide with gas molecules, and that increases its temperature. Temperature inside a vacuum is kind of a strange thing, but that's the effect. So these peptides are carrying protons on board because those were deposited in the process of ionization. As we increase the temperature of the peptide, the bonds become unstable because the proton starts moving around, and that creates fragment ions. Typically, in collision-induced dissociation, the name for this process, we see that a peptide is breaking at peptide bonds, and the N-terminal part of it, the left end of the peptide, breaks to form a B ion, and the C-terminal part of it, the right end of the peptide, forms a Y ion. And we number these based on how many amino acids they contain. If it breaks at the second peptide bond, then you have a B2 ion because it contains only 2 amino acids. And the Y ion contains the rest of the amino acids. So if you have an 8-mer that breaks at the second peptide, you have a B2 ion and a Y6 ion. So tandem mass spectrometry measures the fragments produced by breaking a peptide, not the peptide itself. That's already been measured in mass spectrometry. Tandem mass spectrometry is a list of the fragments that result from breaking a particular peptide. Okay, so how does that play out in the real world? In this case, we have a 10 amino acid peptide, a decapeptide. Now we see that I've drawn a red line in there after the 4th amino acid. That is to say, we are breaking the 4th peptide bond in this 10 residue peptide. And we see that the N-terminus would then form a B4 ion, and then the last 6 amino acids form a Y6 ion. So we can see that they appear in the tandem mass spectrum that results from it. The 415.2 ion represents the first 4 amino acids, the sum of their amino acids plus the number of ionizing protons. And the Y6 ion represents the last 6 amino acids, thus is heavier than the the B4 ion. Plus we add water to get the mass of the Y ion. So we see then that this breakage at one peptide bond has created 2 separate fragments that both appear in the tandem mass spectrum from this peptide. Now obviously reading this spectrum to get the sequence would be very difficult, but we have a lot of software that helps us with that, and that's what the next lecture is about. Now gels have played a very important role for a long time in proteomics, and I don't want to completely ignore them. So in this this image, we're showing a whole cell lysate of E. coli, all of the proteins of E. coli that are being separated on one dimension, and that is the molecular weight dimension of this. So we've stretched out the proteins, caused them to become charged negatively by a detergent. We then apply a charge to the proteins, and they move through the gels based on their masses. So the smallest proteins, again, can wiggle their way through the gel faster than the largest proteins. And we get this separation then of the proteins on that axis. Now that's great if you're working with proteins. It's not so good if you're working with peptides, so we have to keep that in mind. We frequently, though, add a second dimension to use two-dimensional gel electrophoresis. So if we are to separate the proteins based on their isoelectric points, that is the pH at which this protein no longer carries a charge, we can separate them on the pH dimension and then push them into the gel. That creates a two-dimensional map of where these proteins appear in a molecular weight and isoelectric focusing pH axis. So this gives us the ability to separate very complex mixtures of proteins in a gel format. And one of the very useful things that has come out of this is the ability to detect particular antigens from cells. So the Western blot represents our way of using antibodies that have been labeled in some way, maybe by fluorescence, maybe by radioactivity, to label, to find where on this 2D gel map is the protein to which this antibody reacts. So Western blotting is the answer to that. Now, Western blotting has long been considered kind of the gold standard for how you prove that this protein you're claiming is really present. And yet, there are a lot of weaknesses to this. What if you can't get an antibody that is specific to this protein of interest? So a lot of folks are trying to move away from the use of antibodies to use mass spectrometry instead. Now, the experimental methodology I showed you before is about shotgun proteomics, about a discovery. We don't know what's in the sample, let's find out. Targeted proteomics is quite different because its goal is to ask, what is the quantity of this particular protein that is present in the sample? So tandem mass spectrometry using triple quadruple mass spectrometers is starting to move into this space, and people are less demanding that you must have a Western blot if you've already done the targeted proteomics experiment. So tandem mass spectrometry has become the dominant way in which we identify proteins. Starting in the 1990s, people really started saying, this is the thing we have to do. And we can now detect tens of thousands of peptides from a sample. In some cases, 100,000 different peptides can be identified from a single sample. That's pretty astonishing. Gel electrophoresis has been essential in proteomics, but I have to say that the amount of data we yield in a typical proteomics experiment with mass spectrometry is much, much greater. And so we see that there's kind of this balance that people are shifting away from the easy use of gels to the more complex use of mass spectrometers. But certainly the analysis can be quite a lot more difficult. So measuring proteins is essential to understand the ultimate products of gene expression. You might think that you know how much of a protein is there because you've done a transcriptomic experiment. But the number of copies of mRNA that exist for a particular gene does not map directly to the amount of protein that's present. So proteomics and transcriptomics have side-by-side roles in our understanding gene expression.